Now we turn to Dr. José Antonio Neme Castillo, researcher at IMAS UNAM. He specializes in anomaly detection and bioinformatics, building bridges between classical statistics and modern deep learning approaches. Today, he'll show us how anomaly detection is transforming biomedicine, opening new possibilities for early diagnosis and personalized treatment. Please welcome Dr. José Antonio Neme Castillo.
Thank you. Thank you very much. I'm super happy. I'm thrilled to be here. I would like to thank the organizers, mainly Jessica Huelgas and Elena Gomez. I'm very, very grateful with you with you both because of this invitation. So in the next 45 minutes or so, I'm going to talk about what the title says, anomaly detection in biomedicine. First, I'm going to talk about what are anomalies, and then I'm going to walk you through some projects I have been involved in the last several years now. And well, without further ado, I will start my presentation. First, thanking you all for being here or in the internet. So you don't have to wait until the end. If you have questions, you can interrupt me at any moment. It is better for me, but probably it is better for the organizers if you wait to the end. But if you have any question or answers, as it will be the case now, then please just raise your hand. So you are looking here at the west, at West Mexico City. This is to the west, not too far away from here, some 10 kilometers from here. And I would like to ask you the following question. Do you see any kind of anomaly here? Even though you might not know what an anomaly is, the question is rather simple. What is the anomaly here, if there is such a thing as an anomaly here? Please. Yeah, okay, so the answer is already there. Some streets are like a grid, the Roman way to build cities, and some are not straight. You can see here this street, and actually this street is quite peculiar because the name of this neighborhood is Hipódromo, which means the place where you go to see horses running in the circle. I forgot the name in English. How is it called, the Hipódromo? Racetrack. So thank you. The racetrack is still here. So that is an anomaly. The important thing here is, what did you do? What did your brain perform in order to detect this as an anomaly, or perhaps this special roundabout as an anomaly? We are interested in answering that kind of questions. How do we detect an anomaly when nobody is telling us what to look for? So I started this slide asking you a direct question without describing what is not an anomaly. So in that sense, somehow you figure it out that there was some peculiarities and you were looking for those peculiarities. Another possible answer will be the green parks. So this is Parque México and Parque España. So this is compared to the rest of the data set. These two are anomalies because they are quite green. So in that sense, under that assumption, this can also be anomalies. This is the next one is an easy example. Which one is the anomaly and why? There might be more than one answer depending on the considerations that you take into account. So since I don't have that much time, probably one of the anomalies will be this one, because the composition is not focused on people. But the reason why I put this collection of paintings is this one. This one is by Botticelli. This is the Venus de Nilo, the Venus de Botticelli, sorry. And this is the anomaly because all the remaining paintings were created by Jan Vermeer. So in that sense, this is the anomaly because it is not following the style that is present in all Vermeer's style, Vermeer's painting. So in that sense, this is the anomaly. Again, the important question is how do we know what to look for in order to identify something that doesn't resemble to the rest of the observations? That is what we are going to refer as an anomaly. An instance, a vector that does not resemble the rest of the observations within a data set. But the important part is that nobody is telling us what to look for. We don't know what is creating or generating the observations. This case is quite simple. Do you detect any anomaly or anomalies? Would you mind to tell me which one are the anomalies? Just please. So that one. And is there any other anomaly? Well, this is an anomaly, and this is also an anomaly. The reason is that they don't fit the definition of this spiral. So once you know the generative process, the function that is generating the data, it is straightforward to verify what observations do not fit the generative function. So in that sense, this is a quite straightforward way to detect anomalies. Unfortunately, that is almost never the case. We don't get to know the function that is generating whatever we are observing. That can be any phenomena or procedure, and we usually are not that lucky to just identify that particular mechanism. So in order to detect anomalies, since we don't know the process that is generating the data, we have to infer some properties in an unsupervised way to identify a criteria that will allow us to compare all the observations and identify those that do not fulfill with this criteria, this comparison criteria. And for that, we have to remember that we have a lot of vectors and a lot of dimensions. In general, we have high dimensional spaces. And if we were so lucky as to have an external label or class in which somebody will tell us, this is an anomaly because it's red, this is not an anomaly, this is an expected or usual instance because it's green, and this is an anomaly, and so on and so forth, then the problem will be straightforward. It will be a supervised classification task. We will just train a model, any kind of model, and just find a function that relates these elements in this set with elements in this set. That will be straightforward, but again, that is almost never the case. We are not told in advance what objects are anomalies. So we have to infer that by ourselves. That means by some algorithms. So can you pinpoint the anomaly here if there is an anomaly or more than anomaly? Some of you know the answer, I know, but perhaps it pops out that vector C is a kind of anomaly. The reason might be that it is kind of isolated from the rest. So in that sense, that could be an answer. In order to verify that, what I'm showing here is as an extra dimension coded as the area assigned to the vector. This area assigned to the vector is coding the distance from the vector to its closest neighbor. So you can see C is quite fat now because its closest vector is L, and there is quite some distance from this vector to this other vector. So what we are doing here is characterizing the data set from observations that we already have. And well, it is more obvious now that C might be an anomaly because, well, C is fatter, and that is coding a particularly relevant feature. Another possibility will be to define a neighborhood around this vector, or around each vector. I'm only showing 5 of them. And we can inspect the neighborhood around each vector, the neighborhood for each vector, and we can compute the density. So you might see that almost all vectors, 17 out of 19, have 2 or 3 vectors as within their neighborhoods, with the exception of C, which has 0 vectors as neighbors, and F that has 4 vectors in its neighborhood. So in that sense, vector C and vector F are anomalies. Once again, we are not told the criteria to identify the anomalies, so we have to infer it somehow. Sometimes we infer that criteria from statistics, sometimes we infer it from geometry. So it is an open question, and I will show some other examples. Again, this is the same data, but this data has been embedded, has been transformed to a new space. The new space in the x-axis is the expected distance from the vector to the rest of the vectors, and the y-axis is the maximum distance between a vector, the distance between the vector and its most distant vector. So again, it is the same data as before, but look at this. If I were to ask you the same question, do you see any anomalies here? What will be your answer? Probably the same as before, but it is easier now to answer my question. Is that so? It is quite straightforward now that we have this embedding to answer the question that we are interested in. So in that sense, I like to think of myself as somebody that is not always successfully, but developing algorithms that try to make sense of the data. So what do you see here? Some of you know the answer, so don't answer. So I'm looking at you. You know the answer. What do you see here? It's kind of difficult to make sense of this because we don't have the right tool to inspect into the data. So if we were to have a cylinder, this kind of cylinder, we will be able to detect the relevant patterns that we are seeking for. So in that sense, as I was saying, I like to think of myself as somebody that is somehow trying to develop these kind of algorithms. As I said, not always successfully, unfortunately, but well, we have to try. And the idea is that, well, we want to identify relevant patterns in biomedicine, which is the title of this event and of this talk. And what we have been doing so far is mapping the data from one space to a different space in order to make our lives easier. In some areas of computer science, for example, the ones I work in, we are lazy people. We want to make our lives easier in terms of computational capabilities. So in that sense, we are always trying to transform data to an easier representation. And in that case, we are always trying to make embeddings or mappings. Embeddings is a quite new fancy word, but this is mathematically a transformation. We are converting data from one space to another. So this is a quite convenient embedding in this part of the country, in this part of the city. Since we have the rain season at full strength, we need these kind of things quite often. And well, when we are doing this kind of transformations, we are making our lives easier. So you see here a collection of points defining a circle with just 2 coordinates, the x and y axis. And what you see to the right is the same data set, but with a new embedding. We transform the data in such a way that the y-axis is the expected distance from each point to the rest of the points in the y-axis, in the x-axis, I mean, and in the y-axis you have the distance from each point to its most distant point. So what do we learn with this kind of representation? We get to know things that weren't straightforward in this original representation of the data. So in short, what we are trying to do is to transform data in order to detect peculiarities in an easier fashion. At the end, we want to identify data points, vectors, or observations that do not resemble the rest. So in this case, it would be hard to say that there is an anomaly because all the observations fit perfectly this curve. So in that sense, this is showing us that, well, there are no anomalies in the data. So there are many ways to detect anomalies. An anomaly is, once again, something that doesn't resemble the rest of the observations under an undisclosed criteria. We have to infer that criteria. The basic thing to do is to apply, for example, Grove's test. You just assume that your data is unidimensional, univariate, and it is described by a Gaussian distribution. And if that were the case, then you don't have to do anything else, but just apply the Grove's test, which basically tells us the p-value. And you might remember from your statistics or your statistics classes, lectures, that the p-value is the probability that something as extreme as what you are observing was found or detected by chance. So in that sense, this is interesting in some areas, not too interesting for me, so I will just skip it to the next relevant statistical method to detect anomalies. It is called the seed score. In Spanish, you might refer to it as unidad tipificada. The idea is that you want to inspect the data and obtain first order statistics, for example, the mean, and then inspect again the data and obtain a second order statistics, for example, the standard deviation, and then you will inspect the data a third time, and you will compare each observation with the mean, and you will weight it by the standard deviation. So the final result from this analysis is that you will, for each observation, you will have a seed score that tells you how different is the observation I from the mean, how far away it is from the mean. This is quite common in statistics. Again, this is more interesting than the Grove's test. You are observing here pollution data from Mexico City, real data, and you can observe that the dots that actually represent days, the days in red, are anomalies because they have a very high set score. If you want to elaborate a little more to make things more interesting, you might define something that is called modified set score. So in this case, you are not using the mean, but the median, and you are characterizing the data in a more interesting way, but still it is quite straightforward. So I'm moving on to more interesting algorithms. As I was saying, the idea is to define or to infer a criteria for comparing data and detecting those instances that do not resemble the rest of the observations within our data set. And we can do that, of course, in a supervised way, which is, as I described a few slides ago. This will be possible if somebody could tell us if the data, if any particular data is an anomaly, for example, or an expected value or a common value. In that sense, we will just train a model, and that will be it, not too much. But since nobody is giving us the label, the external label, we have to infer it by ourselves. And of course, we can do that by means of unsupervised learning, which is more or less inspecting the data to try to find something interesting there without any external label. It is at the end creating a function. That function might be anything from linear regression to deep learning architectures, but the idea is that we want to assign a label. The label is this vector is an anomaly or this vector is not an anomaly. So without further ado, let me introduce you to the first method that helps us to detect anomalies. It is quite simple as well. The idea is that you have a collection of points and you aim to infer a certain structure in the data from, again, defining neighborhoods. You might see here that for each point, there is a neighborhood. And if this neighborhood is overlapping, for example, the neighborhood of vector E with the neighborhood of vector C. Then if they are overlapping, you just connect them with an edge. And at the end, you create a graph. You can see there is a graph here, and you can detect that there are some points that are not really part of that graph. So at the end, we are applying geometry. I'm skipping a lot of steps, very interesting steps, of course, but the idea is that these vectors are isolated. And since they are isolated, they are anomalies. Our criteria here is that a vector is not part of the graph, then the vector is an anomaly. Now we have our first criteria to compare vectors and to decide whether a vector is an anomaly or not. There are some other methods that are also very interesting. To the left, you can see isolation forests. The idea is that you have data and you want to isolate each vector. That means that you want to randomly create areas in the vector so that each vector is the only one in that area. So for example, if I were to isolate the red vector here, I can start by randomly deciding to cut the space here. And now I have two regions. And this vector is not isolated because there is another vector here. And then since it is not done, since I haven't isolated this vector, then I have to continue with this process. Then I might decide that I'm going to cut this vector here, the space here, and always trying to randomly generate hyperplanes that try to isolate the vector. So the idea in isolation forests is that the harder it is to isolate a vector, the less its level of anomaly. That means that the easier a vector is isolated, then the higher its anomaly score. That is the criteria. Now we have a different criteria. Before we have the concept of a graph, and if something is not part of a graph, then it is an anomaly. Now the idea is that if a vector is easily isolated from the rest, then its anomaly score is high. So that is another way to look at anomaly detection criteria. The third one is also quite easy. It's called local outlier factor. The idea is that I want to characterize the neighborhood of a vector. Once I have that characterization, I will compare it to the characterization of the neighbors of A. So I will just somehow compare characterizations of neighborhoods. And if that comparison gives me a high score, then the vector is an anomaly in local terms. And at the end, it is quite easy as well. This method, local outlier factor, is derived from DB-scan. The authors are more or less the same group of people in Germany, actually. So the idea again here is that in order to decide if something is an anomaly, we have to characterize its neighborhood in terms of density or some other criteria, and then compare that characterization with the characterization of the neighbors of that vector. So in that sense, we are comparing neighborhoods. There are many other ways to detect anomalies. This is quite interesting. Do you recognize this picture? This is Blade Runner from Ridley Scott, 1982. You can see Rachel here at the card here. And what you are looking here is a autoencoder. Basically, the idea of an autoencoder, this one is based in deep learning architectures, is that we have a high dimensional space defined by the size of the frame and the colors of the pixels. This is a very high dimensional. The network learns to compress it, going from a very high dimensional space to a very low dimensional space, but then the network learns to decode this information, trying to reconstruct the input image as similar as possible. You can see here that this image is not the same as this one, but it is quite surprising that we go from a space of 1 million dimensions to 2 dimensions, and then back again to 1 million dimensions. It is, for me, this is still quite amazing that something is compressed so much and then decompressed as to have something very similar to this one. So this is quite useful, a quite useful tool in many applications, biomedical applications and so on. But we are using this kind of networks and some related to this one in order to detect anomalies. Yes, please. Actually, this was a 250 seats plus the 3 channels, red, green, blue.
Do you have a metric for the complexity of the do you have a metric for the complexity of the two-dimensional space compared to the larger space?
Yes, actually, we computed the entropy for this the whole set of images, and the compression rate was quite high, but the error was quite high as well for this particular case. But for some other applications, the error is quite low, as I will show you in a few more slides. Thank you for your question. Quite interesting question. Thank you. So basically, again, we are trying to detect anomalies. And what would you think it is a good way to identify anomalies with this algorithm? Well, since I don't have much time to wait for your answers, I will reveal it right now. So for each vector that you are analyzing with this kind of networks, you get an error, meaning the comparison between the output and the input. The higher the error, well, you will go up here, and suppose you have all the frames for this picture or whatever data you are observing, you have all the observations here, and you assign to each observation, you have error measure, which basically computes how similar the input and the output are when compared under certain criteria. And the comparison criteria will be this, exactly this and this. Those vectors that are very hard to reconstruct or that are very easily reconstructed are the anomalies. That is the criteria under this kind of architecture, compared again to the densities or to the belonging to a graph or not. The criteria here is quite different and I think quite interesting. The harder a vector is to reconstruct, then the higher its anomaly score. Or on the other way, if a vector is easily reconstruct, then it means that it might be an anomaly. Again, the idea is that an anomaly is something that doesn't resemble the rest of the observations under a certain criteria. And since we don't know that criteria, we have to think of ideas of how to compare things. So in biomedicine, these ideas, the ones that I just depicted, are quite easy, have been heavily applied. So this particular application is quite interesting. This network is actually depicted here, is trying to detect physical activities that may lead to cardiac arrest. So the idea is that nobody is giving an external label of whether a certain activity pattern led the person to cardiac arrest or not, but the method was trying to infer from the data based on a network similar to what I just showed you. The method is trying to detect anomalies, and those anomalies are indeed activity patterns that later were known to belong to people that had some cardiac problems. And well, there are many other applications of what I just said. For example, in ophthalmology, there are several descriptions of several images, sorry, that have not been labeled by the specialist. And this method from hand to a couple of years ago was able to detect images that are peculiar under certain considerations, and those considerations are the reconstructing error of the network. And there are quite many. This is also from ophthalmology. You see, you observe here the back of the eye near the macula, where the optic nerve is located. And again, these methods have been useful. This is for Alzheimer from imaging, and quite many other applications. I'm just quickly going to talk about a couple of projects I have been involved in. The first one is related to genomics. And actually, in this project, we have developed the concept of anomalous expression. In general, when you work with bioinformaticians, they tend to do something called differential expression. So, for example, a gene that is in condition 1 is characterized in terms of something. So you can observe here the expression of this gene. The expression means you can think about it as the number of copies that are present of a gene. And you can study the same gene in a different condition. For example, this is healthy tissue, this is from a tumor, or this is a person, and this is a person after ingesting vitamin D, which is the project behind this. And you want to actually compare these two situations for the same gene. The question you want to answer at the end in this kind of analysis is whether there is an important change between these two conditions and in what genes that change is detected. So the common way to do it is to apply statistics. So from this you infer a probability distribution for condition 1 and for the same gene you obtain a different probability distribution for the second condition, and then you compare these probability functions. You can compare it by many using several tools, for example, Kolmogorov-Smirnov test, or a binomial test, or negative binomial test, or many others. But it is quite challenging to reach an agreement, because there are several small steps that have to be taken into account. And at the end, I'm showing here a lot of replicates, as it is called, this kind of doing the experiment again and again and again. And it is quite expensive to do these experiments, and to infer a nice probability distribution is not straightforward. And since it is not straightforward, there are several algorithms making several assumptions, statistical assumptions from the data. There are quite many of these tools. And well, since there are already quite many tools, we ask ourselves, hey, can we make more noise and propose a different tool based on a different set of ideas? And Professor Lee at the University of Southern California was very she was one of the first researchers advising or telling the people that something might be wrong if we don't have an agreement of how to detect genes that are changing when something is changing. So, for example, if you suffer from some illness, some genes might be expressed in a different way. But the answer that each of these methods gives you of what are those genes that are behaving differently is not unique. So that means there is no consensus, and since we are basing a lot of our medical research in this kind of algorithms, Professor Lee asked herself, well, probably we are doing something wrong if we need to create that many more algorithms to try to identify those genes. And of course, we ask ourselves, can anomaly detection be of some help here? And that is what I'm going to talk about in the very next slides. So for that, allow me to talk about compositional data. Compositional data is quite interesting a property of some data. For example, you can see food here. And when you think in any kind of food, your favorite food, for example, carnitas or blackburst or whatever, you can always define any particular kind of food in terms of its protein, carbohydrates, or fat content. The idea is that for any kind of food, the sum of its components adds up to 100% or to 1 if you are talking about the relative frequency. So the idea is that no matter what food you are thinking about, this criteria, this feature is present in all the data. The sum the addition of all its components adds up to 100%. That means that for 3 variables, 3 dimensions, you can define in this probability simplets, you can, each point here represents some kind of food. So for example, beans here are also here. Where are the beans? So it has almost 40% protein, 37% of 27% of carbohydrates, and the rest is fat. So all the data is located in these simplets. So with this information, we can think of our replicates in the gene expression as points in these simplets. And you can observe here that all the points in for 3 dimensions will be located in the surface of this structure. It is called the probability simplets. And here we can apply a lot of interesting algorithms relying on geometry. In particular, something called information geometry. If you don't know the concept, please go after this talk and get to know more about information geometry. Is quite a fascinating subject. But the idea is that we want to represent each gene as a point in these simplets. And then once we have that, we have to ask ourselves, well, how do we compare the position of those genes in that probability simplets? The answer is not straightforward. It will be as equivalently as as this problem. I want to walk from here to Biomedicine, which is another institute located not too far away from here. But if you walk through this path, you will more or less tire your legs. But if you were able to fly, you used to follow this line. And if you actually follow the path, the trail that goes from here to there, you will take this line. So the answer is, how far away are we located from Biomedicas? Well, it depends on the geometry, and of course it depends on the distance function that we are applying. So saying that, for example, this triangle is the probability simplets. I'm just showing here some rings, not algebraic rings, but geometric rings. You know, a ring is the superposition of circles. And if we were to apply the Euclidean distance, this is what we will obtain for a set of rings. If we were to apply this Wasserstein distance, which is a rather important distance, and I'm going to talk about it in just one slide, this is the set of rings that we obtain. And if we apply the Aitchison distance, this is the set of rings. And if we continue doing so, and we apply different distance functions, we obtain quite different things. So what is the right distance function to apply? Which one would you say is the one that we should apply? Well, the answer, it is almost never easy. It depends on the geometry that you want to focus on. And well, I can continue with this, but the idea is the following. Remember, we have a probability simplets. In terms of food, this was, remember, protein, carbohydrates, or fat. In terms of expression, this was the expression in the first experiment, expression on the second experiment, expression on the third experiment. Now suppose something different. Suppose there are 3 age groups from 20 to 30 and so on and so forth. And there are 3 illness. And this is representing the prevalence of that illness for each group, age group, for each age group. Since most of you here in the conference room belong to this group, probably you will be more interested in illness B, because illness B is mainly affecting people in your age range. So this will be the illness that will affect you the most. So you should be worried about that. But now, allow me to ask you the next question. What will be the second most prominent illness that you should be aware of or worried about? See? It depends. Actually, that's the correct answer. But it depends. If you think of this structure in terms of Euclidean geometry, if you think this is a point with coordinates 1, 0, 0, 0, 1, 0, and 1, 0, 0. If this were to be an Euclidean space, then the answer will be, it doesn't matter. Either C or A will affect me equally, or will be as close to me as they both will be equally distant to me, or I mean to the point B, because the distance from here to here is the same than the distance from here to here, if you are assuming an Euclidean geometry. But since we know that there are other geometries, then we can remember that what you just said is important. The second most prominent illness for this age group would be this one. Even though in this space, it seems that both A and C are at the same distance, they are not, because, well, intuitively, this is closer to your age group, since from 31 to 50, it is closer than 51 to 70. And the whole idea of this is condensed in something called the Wasserstein distance. And the Wasserstein distance, also called the water current distance, it is solved by something called optimal transport. So the idea of optimal transport is you have a collection of things, you want to move things to achieve a second distribution. There are many ways to do so. So for example, you can move this point to this point, this point to this point, this point to this location, and this one to this location. And the idea is that you want to do it in such a way that the movement of things is as small as possible. That is why it is called optimal transport. And the ideas come from Gaspard Monge in France in the 19th century. But just try to picture this. You want to move the blue points to coincide with the red points. So you want to convert, to transform an A into an L. What would you do? Well, you can do it, you can do a lot of possible movements. You can see two of such possible movements here. The one in the left is achieved by optimal transport. So the distance, the Wasserstein distance between L and A is given by the addition of the local displacements. So I have to add up all these movements, and that will give me the Wasserstein distance of two things, the L distribution and the A distribution. And of course, I can do it some other way, but the important thing here is that this sum is higher than this sum. So in that sense, you might remember that any distance function has to fulfill three criteria, and if you want to call it a metric, then you have to add a fourth constraint, right? Everybody remembers that? Okay, good. So if you want to learn more about that, read this marvelous book from Cedric Villani, field medalist. And the idea is quite simple. If you want to learn more about distances, which I always recommend to my students, read this book. This is an encyclopedia. It is almost 1,000 pages. This is a beautiful book describing a lot of the concepts needed to understand the concept of distance and families of distances and when to use what distance and so on and so forth. Going back to our data, to our case of gene expression, and I am almost finishing, we want to identify genes that are behaving differently in two conditions. We don't want to apply only statistics for that, which is almost always the case. What we want to do is to convert the expression of each gene into a point in the simplicial complex or the probability complex, and then we want to do it the same for the same gene, but in the second condition. And then what we do is to compute the distance, the Wasserstein distance of how much this gene moves from one condition to the other condition. And remember, we want to do it not in an Euclidean way, but considering a different kind of geometry. So we are just counting or just computing the distance that a gene traveled within two conditions. That's all, that's the whole idea. And at the end, we do this for all genes that are under study. And here we apply statistics. In this part of the analysis, we just count the number of genes that traveled such distance or any given distance. And here we can observe some patterns, as you might see, you will see in a few more slides, in a couple of slides, that some genes, genes might have been characterized by a very large distance. That means that they moved quite a lot from one condition to the other, which is more or less what is observed here. This is one of my former students, Michelle Mata. This is from her thesis work. And this is the important thing here. What we were, we were analyzing data, human data, quite sensitive data, from a group of people that ingested vitamin D. I collaborate with some colleagues that are interested in the effect of vitamin D in human health. And that is quite difficult because, well, there are some chemical reactions that have to be present in order for the vitamin D to get active. And the idea here is that we apply different methods that detect relevant genes that change their expression within these two conditions after ingesting vitamin D and before ingesting vitamin D. These two methods, DSEC2 and HR, are quite common methods. And what you observe here is more or less a Venn diagram. You can observe here that there is not a consensus. It is quite difficult to really identify those genes that are behaving in a different way in two different conditions. Our method, we apply these two methods, and these two methods are based in anomaly detection. And actually, when we investigate the ontology of those genes that were selected, the ontologies are more related to what we know is vitamin D is doing. So in that sense, it was a success. I'm not going to talk much about this, but this is another problem in biomedicine, actually in SARS-CoV-2, the genome of SARS-CoV-2. We analyzed the genome, actually the codon usage of a genome, which is a characterization of the whole list of letters. And the important thing here is that you can, this is the characterization of the first genome of SARS-CoV-2 isolated in Mexico. It was isolated not too far away from here, and this is the one that was isolated later. And you can observe here, this is a characterization of the genome of SARS-CoV-2, and this is a characterization of another, the same virus, but in another person a year later. And basically, we applied anomaly detection algorithms to this kind of characterization to public data, and we observed something peculiar. Here you observe the anomaly score. Each dot represents a genome of a virus isolated from a human. And this was quite peculiar. The number of anomalies is not randomly distributed in the country. The vast majority of anomalies are in a state called Puebla, not too far away from here. We applied one method here, and the vast majority of anomalies were from this state. We applied a different method, and surprise, Puebla is again showing the largest amount of anomalies, quite many. We applied some other methods, and again, Puebla is presenting something peculiar. We applied this is from ISOMAP, some other method, and well, we were trying to find out what was happening. And actually, since we have metadata from all these patients, we were able to isolate all these patients in Puebla, more or less in this area. All these people that from which the virus was isolated and sequenced and analyzed live around here. So if you know Puebla, you might know that Cholula is around here. So Puebla city center is around here, if I am correct. And well, the idea behind this is that something was happening. The virus was characterized by something peculiar, and there are two possibilities. Either the virus mutated in this particular area of the country in quite a strange way, or all these people went to the same laboratory and big mistakes were made there. Either case, the method was able to detect that some anomalies were present in the data. It seems to be that it was the second case. We, of course, communicated it to the authorities, and they take a course. So basically, I'm not going to talk much about this, but it has to do with vitamin D. And with this method, we analyzed transcriptomic and epigenomic effects on 45 patients. And you can observe here that some of the patients are kind of isolated. These patients actually do not respond to vitamin D. That means they don't need to take vitamin D because they are not able to process it properly. And this is a quite important thing because vitamin D is a micronutrient that is vital for human health. If you don't take enough vitamin D, then your bones will not be healthy. There will be less density. Calcium carbonate will be lower, and there are many other illness associated to low levels of vitamin D. But with this method, again, applying anomaly detection algorithms, we were able to detect some individuals that after a few days were not responding. So that means they have to, the endocrinologist has to look carefully into their cases. So basically, this is again the same information. I don't want to take more of your time. The idea here, what I have been talking about is anomalies. An anomaly, again, is an instance or an observation that doesn't reflect or doesn't resemble the rest of the observations within a data set. But we don't know how to compare the observations, so we have to infer them either based on density concepts, geometry concepts, statistical concepts. And this is the final slide. Actually, I would like to thank you all for being here. And here are some of my collaborators. I have collaborators here in Mexico, in Poland, Kazakhstan as well. And these are more or less the people that are behind what you just saw, the students. Most of them are from the postgrad here in computer science. So with that, I would like to thank you for your time. And if there are any questions, I would be happy to try to answer them.
Thank you so much, Dr. Neme Castillo. What do you think are the directions, since you are a researcher here at IMAS, what do you think are the next steps in terms of research regarding anomaly detection in the next 5 years in Mexico in order to have a better public health?
Yeah, well, that's a very good and difficult question to answer. First, I always try to convince students from the computer science or mathematics fields to get interested in biology or biomedicine. That is the tricky part. People from biomedicine, they are aware that algorithms are going to help them. And that is very good because after some painful time, they learned some computer science, some programming. But the other way around is not happening. I am always struggling to convince computer science students or mathematics students to get interested in biomedicine. That, I will say, is the most important thing, to convince people, students or colleagues in the mathematical side, let's say, to get interested in these fields. That will be a major success, I'm sure.
To create a bridge.
Yes.
In between, okay. Thank you so much. If you guys have any questions, remember you can raise your hand and we will give you a microphone so everyone can listen to the question. And we would like to thank you as well for being here, especially those who are online. Over 100 people are watching us online and through YouTube and Zoom and the Zoom meeting. And that's everything. If anyone has a question, okay. That would be everything. Thank you so much.
Thank you. Thank you.
Again, an
Thank you.
applause for Dr. Neme Castillo, please.
Thank you.